I want to thank Dimitri and also thank Michael for, awarding us this grant that allows me to come up here and give a talk. So the grant just kicked in, and we don't have much data to show you, but I'm just going to give you an outline of what we're trying to do, and also provide some shared resources that I think would be quite helpful for the entire group here. I want to first thank the multidisciplinary team members that we have. Our team has expertise that covers iPSL biology, multiomics, AI machine learning, biomaterials, biofabrication, and also gene editing. My co-PI, Dr. Afshin Behesti, he has a lot of experience in analyzing large-scale data sets from space flights. I also want to highlight our collaboration with the Space Tango, who will be providing our engineering, also flight hardware support, and also program manager Amanda Chase, and then James, who's been quite instrumental here, and he's also here in the audience as well. And I think Michael kind of mentioned about this here, so I won't talk too much about the background. I think, as you all know, there's quite a bit of space flight health hazards. Exposes the astronauts to multiple stressors such as microgravity, radiation, isolation and confinement, as well as many other physiological stress. Among the physiological stress, I think the microgravity really has a clear and measurable effect on the cardiovascular physiology, as well as inflammation and accelerated aging. And this is the focus of our team here. So our lab has had previous experience with space research related to microgravity. For example, back in 2016, we sent for the first time 2D iPSL-derived cardiomyocytes with the SpaceX CRS-9 launch. And this was actually done by Arun Sharma when he was still a graduate student in my lab, and he's now a PI of one of the projects here. Congratulations, Arun. So here we show that the microgravity can cause calcium mishandling and also transcriptomic remodeling. And then in 2019, we sent 3D iPSL-derived engineered heart tissues with the SpaceX 18, CRS-18 launch, and we show clear evidence of mitochondrial remodeling in these engineered heart tissues. And then in 2023, for the SpaceX CRS-27 launch, we use 3D cardiac organoids as shown here, which offer a nice balance between the structural complexity and also the experimental throughput. And we were able to elucidate the inflammation markers that are quite different between ground control and also flight samples as shown here. And this gives us some hints as to what therapeutic targets that we want to go to, because ultimately we're trying to develop countermeasures that target microgravity-induced inflammation and aging. And so for the UG3, UH3, our project goal here is to further understand the impact of microgravity exposure on cardiovascular homeostasis, and importantly, to identify effective countermeasures. During the UG3 phase, we will develop automated platforms for longitudinal profiling of 3D vascularized cardiac organoids in space. And then during the UH3 phase, we will test candidate countermeasures for spaceflight-induced cardiovascular complications. For AIM-1, this is where we are at right now. In the first 6 months, our immediate goal is to develop an automated platform that allows us to monitor the cardiac system in real time during spaceflight. And to do this, we'll be generating a human iPSL reporter line that expresses troponin T driving EGFP, which reports for cardiomyocytes, CDH5 driving mCherry reports for endothelial cells, and the ISRE and gas at BFP that allows us to track inflammatory signals dynamically. Now, once these cells are edited, we will then confirm the expression response to interferon gamma, TNF alpha, and also interleukin 6. And once that's done, we will then differentiate these cells into 3D vascularized cardiac organoids. Now, for generating these vascularized cardiac organoids, we will use a protocol that we recently published in Science. This protocol gives us a robust and consistent vascularization of the cardiac organoids, as well as the liver organoids. I think importantly, the liver organoids, the vascularized liver organoids allows us to study the first pass metabolism of drugs before we expose them to the vascularized cardiac organoids. Our implementation partner will be Space Tango. Space Tango has a significant expertise in designing self-contained automated units that enable researchers to conduct experiments under microgravity with minimal crew intervention. And so shown here, this is a cube lab. This is composed of 12 customizable wells. And these units are equipped with high resolution phase contrast and fluorescence cameras, enabling real-time imaging and data capture. Now, since one of the goals of this tissue consortium meeting is really to provide shared resources, I would like to introduce to the group here our human iPS cell biobank. This is a joint effort between the Stanford Cardiovascular Institute and also a startup company, Greenstone Biosciences, which I co-founded. Currently, we have generated iPS cells for more than 2,500 patients with different medical history, ethnicity, and sex. This is the world's largest iPS cell biobank. These lines are linked to medical information. We perform multi-omics sequencing on them to help us understand how the human genetic variants impact the drug response phenotypes. We also work with the FDA on drug safety testing, and we have a robust sharing resource plan with the broader scientific community. In fact, today we sent out more than 4,000 bios to more than 400 investigators. Another shared resource we can provide is the various AI screening and molecular docking platforms that we have developed. Here, for example, we could use the FDA repurposed library, the Zinc22 library, the WuXi library, and the Enamine library, or the Greenstone virtual library. We can screen a diverse chemical library against both the target as well as the off-target proteins using AutoDock VINA. The top 50% of these binders can then be further refined in both two. The toxicity profiles can also be screened using the Greenstone AMA AI. This is a free software available on the website. And then we also do the molecular dynamic simulation to give us a multidimensional analysis of the drugs with drug-like properties. This slide highlights the drug discovery pipeline that we typically go at Stanford and also at Greenstone. That goes from genomics to in silico, to in chemical, to in vivo target validation. Because of time, I won't go into the specifics of each step, but I'll be happy to answer questions during the discussion. Next, I just want to briefly highlight 3 examples of how we use this pipeline here to lead to molecular insights, to target discovery, and also to phase 1 clinical trials. So the first example is shown here. This is a study related to marijuana. I think as some of you may know, marijuana is legal in the U.S. in about 40 states and also 24 states for medicinal use. However, excessive use of marijuana is actually associated with increased myocardial infarction, but the exact mechanism is not well known. And here we show that the psychedelic component of marijuana, the delta-9 THC, binds to the cannabinoid 1 receptor and activates that downstream inflammation. This is the mechanistic angle to explain why patients have increased myocardial infarction. Now, importantly, using AI in silico and also in chemical library screen, we found that ginestin serves as a neutral CB1 antagonist, which means that it sits on the same receptor. It does not activate it, but it prevents delta-9 THC from binding to the same receptor. Ginestin also does not cross the blood-brain barrier, so it does not mitigate the psychedelic effect that the patient wants. And importantly, ginestin is actually an isoflavonol found in soybean. So based on this mechanistic insight, we actually filed an IND application and got approval. This is a phase 1 clinical study by giving ginestin to patients with chronic marijuana to see if ginestin could improve their vascular health. This study is being led by my former postdoc, Mark Chandy, who was the first author on the paper and is now a faculty in Canada. Here's a second example. As cardiac researchers, we know that cardiac fibrosis after MI is bad. It impairs cardiac function, but yet there's no effective clinical therapies that exist. And so as shown in this cartoon here, classically, the TGF-beta can stimulate the canonical pathway, such as the ERK and SMAD pathway, which upregulates extracellular matrix proteins. However, these extracellular matrix proteins can actually further worsen fibrosis by activating another pathway called the MD2-TRL4 pathway. So this becomes our molecular target, and this is the target that we want to design drugs for. So we, again, go through the in silico, in chemical, and library screen. Artesunate came up as a hit. Artesunate is a derivative of dihydroartemisinin. This drug is actually already used in clinic to treat patients with malaria. The scientist, Dr. Tu Yu, who discovered this, received the Nobel Prize in 2015 in medicine. And because artesunate is already an FDA-approved drug, recently we also applied to the FDA and got approval from the FDA to test whether artesunate can be repurposed for treating patients with idiopathic pulmonary fibrosis in a phase 1 trial at Stanford. I will be the sponsor, and Dr. Josh Mooney will be the site PI. So this is another example of combining in vitro, in silico, in chemical, and in vivo studies to quickly lead to a clinical trial, which is the main purpose of NAMS. And finally, here's a third example. As mentioned in the last slide, in damaged heart, the extracellular matrix is a strong mechanical sensing signal that can further promote fibroblast activation by a positive feedback loop. So here we want to look for additional potential therapeutic targets. And we identify a stroma-enriched mechanical sensory kinase called SARC to be a key player. We then perform a large-scale in silico and in chemical docking screen to identify a top candidate of a SARC inhibitor called sirakitinib. So when we combine sirakitinib along with the known TGF-beta inhibitor, we can actually reverse the activated myofibroblast back to a more quiescent fibroblast. And this was published recently in Nature. And so in summary, the goal of our Tissue Chip 2.0 grant is really to understand the impact of microgravity and to develop countermeasures that can mitigate cardiovascular inflammation. Our framework involves using patient-specific iPS cells to help us better understand the molecular mechanisms of diseases. The biological twin data set can then be converted into digital twin data set by multi-omic sequencing. These digital twin data set can then be fed into the AI machine learning softwares that we've developed as well as many others. This would then spit out drugs, and from these drugs, we can then further test it on these iPS cells that we design the drug for, and hence completing this loop. Our ultimate goal is really to develop countermeasures that can mitigate microgravity-induced cardiovascular inflammation and aging. And finally, if you're interested in the topic of drug discovery, I encourage you to register for the 2026 Stanford Drug Discovery Symposium. For this annual meeting, we bring together CEO, CSO, CMO of Big Pharma, biotech, academia, FDA, NIH. For the FDA, I think Beth Hogue and Steve Kozlowski will be there. Venture capital, equity firms, to discuss how we can best accelerate drug development. This is a 2-day symposium. It's actually free for virtual attendees, and I hope many of you will have the opportunity to attend either in person or virtually. And thank you very much for your attention. Thank you.
Thank you, Joe. Are there any questions? We don't really have much time for questions. Is it cannabinoid receptor antagonist or?
Yeah, so I think right now we're looking at the studies that we sent with the 3D organoids from our last launch to look at the inflammatory biomarkers. We do a lot of drug development, so we have to kind of get a sense of which drug the astronauts can take for a long period of time without any side effects. And obviously, I think for the microgravity, people have talked about statins, metformins, and antioxidants, and so forth. Could it be a combination of a couple of these drugs that we can give? That's something that we're looking into. I think the beauty of using these as cells is that we want to look at the heterogeneity that exists, and this heterogeneity came up in the last discussion. In academia, in academia, we don't want heterogeneity because it takes too long for us to publish. In drug development, you want heterogeneity because you can't afford to have 100 million, 500 million, $1 billion of acid that go in, and the heterogeneity kills you because you're betting all on a single cell line, or betting all on one inbred mouse that overexpress some genes or knock out some genes. So here, what we're really interested is, once we're very confident that these are the drugs that we're going to push forward, we will actually test it on a panel of 100 cell lines. These drugs that go, if I have a drug that works in 1 out of 100 cell line, I'm not interested in it. If I have a drug that works in 100 out of 100 cell lines, that means my scientists made up the data. There's no way drugs is that good. If I have a drug that works in 25, 30 out of the 100 lines I have, that's just something that we will go into, and we will push the NCATs and also the NASA and say this is our countermeasure drug. Yeah.
Thank you.